and he will tell us about his work with aptamers and exosomes, or extracellular vesicles. Sorry.
No,
take it away.
That's fine. Thank you. Yeah, I'll actually get into that. Yeah, so before I start, I would like to thank the organizers of the conference to give me the opportunity to discuss some of our work here, especially because about 8 years ago, I was a postdoc in Oxford, so it's nice to be back. Today, I would like to discuss some of our latest work on extracellular vesicles for CRISPR-Cas9 delivery and how aptamers can be helpful for that. So before I start about all the technical details about CRISPR-Cas9, I'd like to start about extracellular vesicles because not everyone is aware of what they are. They are nano-sized particles that are released by cells, all cell types. We haven't found a cell type that doesn't do this yet. And they are bilipid layer particles that are released either directly from the cell membrane, in which case we call them microvesicles, but they can also be released through the endosomal pathway. That process is a little bit more complicated. So what happens there is after endocytosis, you get the formation of this endosome. And so the cell can do this to take up molecules from its environment, and it will eventually fuse with the lysosome, where it will break down all of its contents. So it's kind of like the recycling center of the cell. And on the way there, the cell is going to actively load all sorts of components that it also once shuttled into the lysosome into the extracellular vesicle. So when an endosome forms, the membrane buds inward. So the structure is in a way inside out, but in a way it's not, because whatever is exposed to the cytosol at the cell membrane is still exposed to the cytosol here. But in the formation of these vesicles here, it buds inward a second time. So what you basically have is a particle that's basically been turned inside out twice. So it's back into its original formation. And this structure, which we call a multivesicular body, fuses with the lysosome, but in some cases, and we don't exactly know why, we know some proteins that are involved, it can also fuse with the cell membrane, and these intraluminal vesicles are released, and then they're called exosomes. And together we call them extracellular vesicles. And they play a role in communication between cells, and they can either do that through receptor-ligand interactions directly at the cell surface. But what's interesting is that they can also deliver their contents. So that can be RNA, lipids, protein, metabolites. And in order for that to occur, membrane fusion somewhere needs to happen, otherwise its contents could never end up in the cytosol of the receiving cell. And theoretically, that would be most efficient at the cell surface, but reality shows, or experience shows that that's not really the case, because we block this method of communication specifically when we block uptake of extracellular vesicles through endocytosis. So somewhere in the endolysosomal pathway, this membrane fusion occurs. We think they're interesting for drug delivery because theoretically, because they are biological nanoparticles from human cells, they have the potential to have very low immunogenicity if you were, for example, to use a patient's own cells. They have an interesting biodistribution profile, which is in part determined, of course, by the proteins that are on the outside of the membrane, and they are quite efficient at cargo delivery. Okay, sorry, I just wanted to check that there was nothing wrong with the microphone. Yeah. And so this is what they look like under electron microscope. So this structure here is this multivesicular body. And when it fuses with the membrane, these nano-sized particles are released. So this is a 500 nanometer scale bar. So they're actually quite small, around 100 nanometers. Microvesicles that are released directly from the cell membrane are a bit larger, and they can be up to a micron. So how do you isolate these? Well, there's a lot of techniques, but it starts with cell culture. So you culture cells, and then you can isolate them from the medium. What's important is to culture them in a serum-free medium, because the fetal bovine serum that we usually add has so many extracellular vesicles already in there that it's going to be difficult to kind of separate those from the ones your cells produce. So we culture our cells for 24 hours with Optimum. Then through a few centrifugation steps and a filtration step, we get rid of cells, cell debris, large aggregates, and then we concentrate our sample using tangential flow filtration, where we have a filter which has pores, and everything that's smaller than 100 kilodaltons can go through. And everything that does not go through is basically pumped back into the reservoir. So you keep pumping back and forth, and you're concentrating your sample, and you're getting rid of everything smaller than 100 kilodaltons. And then through size exclusion chromatography, we can actually separate our extracellular vesicles from our soluble proteins. So we start with a quite large volume, 200 to 400 mLs, and in the end, we have half an ml of PBS, which is enriched for our extracellular vesicles. And so in size exclusion chromatography, you can, like I said, separate your large particles from your soluble fraction. And then what you get is a sample like this. And so what you see is extracellular vesicles are quite heterogeneous in size. And like I said, it's enriched. You do also still find some protein aggregates because it's a size-based isolation protocol. So how do you engineer these? Well, there's a lot of chemical tricks you can do, but you can also use the cell to basically produce and load your cargo for you. And so this is a manuscript from a company called Kodiak. The bad news is, unfortunately, they are no longer around. They didn't make it. But the good news is they did publish some of their work, and we can learn from that. And one of their research lines was figuring out, well, how do you convince a cell to load your cargo into an extracellular vesicle? So what they did via ultracentrifugation, they have a density gradient. Extracellular vesicles have a lot of lipids, so they have a relatively low density, so you can separate them from your protein fraction. So this is the protein fraction, these are the particles. They did proteomic analysis, and what they were not only looking for is, well, which proteins are highly abundant in EVs, but then also are actually low at low abundance in cells. So really, what are the proteins that are highly enriched? What they did is they then expressed these proteins with GFP attached to it, to see, well, can we now actively load GFP into EVs? And if you express that in cells, the mean fluorescence intensity doesn't vary that much, but if you look at extracellular vesicles, you see a huge difference. So here on the bottom, we just have cytosolic GFP. You have a handful of molecules per EV, but if you fuse it to the right protein, you can get up to 1,000 molecules per EV. So by just expressing it fused to the right protein, you can have hundreds and hundreds fold increase by a protein that's even produced by your cell. So the cargo that I would like to focus on today is CRISPR-Cas9. I'm sure most of you are familiar. It's a prokaryotic endonuclease that can make double-stranded breaks in DNA. And in order to do this, it requires a guide RNA. And this guide RNA is a bit of a man-made structure because in nature it consists of two separate RNA molecules, a CRISPR RNA that contains the targeting sequence and the transactivating CRISPR RNA. Together, we'll get into that, but they will anneal to each other. And the transactivating CRISPR RNA contains the Cas9 handle. So that's the part that the protein interacts with. So this targeting sequence here determines where Cas9 can make a double-stranded break in DNA. And you can knock out a gene by doing this, but it doesn't do that just because you're making a cut. It's because of the errors that can occur in the repair mechanism. So if the cell repairs this perfectly, Cas9 can cut again, but in some cases it can accidentally insert or delete single or multiple nucleotides, and then of course you get a frame shift because every 3 nucleotides code for an amino acid, and you can truncate your gene or your protein. But Cas9 has way more functionalities, and that started with the production of dead Cas9. So these are 2-point mutations that no longer allow Cas9 to make a double-stranded DNA break. But other than that, it still functions exactly as it normally would. So basically, you can target a genomic region, and Cas9 is going to sit there. It can't make a cut. And by doing this, you can temporarily block transcription because it's physically in the way. But you can also attach specific enzymatic groups. So you now have a tool so you can bring any enzymatic group to the vicinity of a very specific sequence on the genome. In this case, these are called transcriptional activators. These are viral proteins, and they will locally, temporarily induce transcription. What this means is if you target this to a promoter of a gene that's not active, you can temporarily start transcription and thus translation. So you can temporarily express this gene without making any permanent changes in the DNA. Of course, there's also a lot of interest in other enzymatic groups. One of them is a base editor. So here you have an enzymatic group. In this example, it's a cytosine deaminase. And what it does is locally, within the region where you're targeting with your guide RNA, it changes C's into T's. There's even more advanced systems like prime editors where reverse transcriptase will actually use an extra tail that is attached to the 3' end of a guide RNA as a template to locally transcribe DNA to build in specific repairs. So there's a lot of different options. There's quite an extensive tool set. So why am I bringing all of this up? Now, a lot of these are based on enzymatic groups that are attached either to the C or N terminus of Cas9. So if you want to make a modular system to load all these different Cas9 variants, you may not want to use the strategy where you make a direct fusion protein because it's possible that your fusion is going to interfere with these enzymatic groups. So we wanted to get around that and make a modular system, and to do that, we started to focus on the guide RNA instead. So already back in 2015, it was described that there are a few things that you can get away with when making modifications to a guide RNA. So this is the Cas9 guide RNA complex. And here in gray and red, you see the guide RNA. And here in red are two hairpins that are on the outside of the protein. And what's quite interesting is that you can make changes to these hairpins, and as long as they're still hairpins with generally a similar shape, Cas9 still works just fine. And so what's already been shown is that you can replace them with specific hairpins that can then be bound by specific proteins that can bind them. In this case, this is from the MS2 bacteriophage. They have a hairpin which can very specifically be bound by the MS2 capsid protein. And the nice thing about this is you are now binding this Cas9 guide RNA complex without making direct fusions to the C or N terminus. So if you have any enzymes here, you're not interfering with them by any type of steric hindrance. Spoiler alert, most of the time. So we thought, well, can we use this to load Cas9 into EVs? So what does it look like when I'm talking about these modifications? So this is the general shape of a guide RNA. You start with your targeting sequence. And this tetraloop, this basically is a man-made structure because originally we had a CRISPR guide RNA that had a repeat and a transactivating CRISPR guide RNA that had an anti-repeat. But we just turned that into one large hairpin called the tetraloop, so you can express a single molecule. And then at the end, there's stem loops 1, 2, and 3. But this tetraloop and this stem loop 2 are actually sticking out of the Cas9 protein. So you can add these MS2 aptamers on top of these original hairpins, and everything still works. So here was our idea. We had CD63. It's a protein, it's a tetraspanin. It's highly enriched in extracellular vesicles, specifically in exosomes. And on the N-terminus, on the intraluminal side, we expressed two MS2 code proteins because two of them bind one hairpin. And our idea is we're going to co-express Cas9 and these modified guide RNAs. If they form a complex, these MCPs can bind the hairpins that stick out of this complex and drag the entire thing into an extracellular vesicle. So does this work? Yes. Ooh, that's hail. If you express this construct, it doesn't matter, it doesn't really have an effect on the amount of Cas9 in your cell. But if you look at extracellular vesicles, this is without the loading construct, this is with. So we see a strong enrichment of Cas9. And we see the same for the guide RNA. This is the guide RNA within the cell, this within the extracellular vesicle, and you see a strong enrichment. And we confirm that by qPCR as well as digital droplet PCR. Still, though, with this enormous increase with digital droplet PCR, we see that we have about one guide RNA per 20,000 EVs. So that is way better than we had before, but definitely one of the limiting factors. However, this still presents a problem because Cas9 needs to be able to go into the nucleus of the target cell. And right now, this is quite a strong interaction. Everything is still stuck to the membrane. So we thought, well, we need to come up with a trick to facilitate release. And we found this paper. This is a photocleavable domain. It's quite an interesting one. It's derived from M-maple. So M-maple is a fluorescent protein, and normally it's green fluorescent. If you activate it with lower wavelengths, around 400 nanometers, it undergoes a conformational change and becomes red fluorescent. But in this paper, they found some point mutations where this red fluorescent conformation is unstable and the protein breaks. So what you have is a green fluorescent protein that you can activate with higher range UV light, and all of a sudden this protein breaks. So what we did is we cloned that in between the membrane protein and the RNA binding domains. So of course what happens is, after isolation, you can treat them with 400 nanometer light, this breaks. The RNA binding domains are still attached to the guide RNA outside of Cas9, but everything has come off the membrane. So first, to verify whether this worked, we expressed this protein, we added an HA tag, and indeed, so this is the full-length protein. It has multiple bands because it's a heavy glycosylated protein, so there's always a bit of a smear there. But the moment that we start treating these cells with 400 nanometer light, you see that it cleaves into two components, and we see that both within the cell and in isolated EVs. So that works quite nicely. And then we thought, well, how are we going to easily screen all our different constructs for Cas9 delivery? So previously, back when I was a postdoc in Oxford at the lab of Matthew Wood, we made a reporter system for Cas9 activity. And it works as follows. You have M-cherry, a red fluorescent protein, which is expressed. And then we added a few amino acids at the end that don't really do anything. They also don't interfere with the function. followed by a stop codon. So if Cas9 creates a frame shift here, the stop codon is going to be out of frame, and we have open reading frames for GFP, 1 or 2 nucleotides right behind there that could come into frame if you have these frame shifts. So here's what that looks like. Normally our cells are red fluorescent, but not green fluorescent, but if we transfect them with Cas9 and the correct targeting guide RNA, they will permanently start to express GFP at a very high level. So we transfected our cells with our constructs. After 24 hours, changed the medium to optimum. After 24 hours, isolated our EVs, added them to the stoplight cells, and saw nothing except for a positive control, which was plasmid DNA transfection. So we were happy to see our reporter work, but everything else was a little bit disappointing. So we went back to the drawing board and thought, well, what could still be a limiting factor? We have loading, we have release. And it was already actually mentioned during the first talk today. And the culprit in this case was endosomal escape. So of course, endosomal escape, we know that somewhere in the endolysosomal pathway, extracellular vesicle-based delivery occurs through membrane fusion, but it's not known exactly which proteins are involved. So we thought, well, to study if this is a limiting factor, we'll add a viral protein, VSVG. And what this protein does is it facilitates membrane fusion the moment pH drops. So it undergoes a conformational change, grabs the nearest membrane, pulls it back in, and facilitates membrane fusion. So when we co-express this, everything worked like a charm. We saw a lot of green cells, so that means a lot of DNA editing. What was also really nice to see is if we did not treat our EVs with 400 nanometer lights, so if Cas9 was still stuck to the membrane on the inside of the EV, even though you had membrane fusion in the endolysosomal pathway, you saw very little delivery. But if we first pre-treated it, we saw very efficient delivery. So we wanted to publish this, and we said, clearly it's endosomal escape. And one of the reviewers said, prove it. So what we did is we labeled Cas9 with M-stay gold, which is a very bright green fluorescent protein. And then we added them to cells that were stably expressing BFP-tagged histones. So their nucleus is a blue fluorescence. And after 1 hour, we didn't really see a big difference. But after 4 hours in the cells that were treated with EVs that also were expressing VSVG, we started to see this green signal within the nuclei. And without VSVG, we did not. And we can actually measure that if we look at MFI per nucleus. So our conclusion was indeed that VSVG facilitates endosomal escape, which is necessary for delivery to the nuclei. So like I said at the beginning, there's a couple of different proteins that are highly enriched in EVs, and this is an entire field of research. So we picked a few of the most commonly mentioned ones in literature at the time. So CD63, CD9, CD81 are all tetraspanins from the same family. Myristylation, a post-translational modification, and one that was specific for microvesicles. And what we found is that your choice of protein makes a huge difference. And we saw that specifically CD9 worked well. It gave twice as much delivery as a CD63. And to determine what was the cause of that, we started with looking at loading efficiency. And indeed, CD9, twice as much Cas9 in the EV. So it's not that the cells are treating these particles differently. It's really the amount of Cas9 that you can get in there. And so we saw this at multiple dosages. And of course, it's a dose response game. So if you go high enough in your dosage, we could get up to 60% of our cells expressing GFP. So that worked quite nicely. And then we thought, well, we started off saying we want a modular system that we can use to deliver different types of Cas9. So we started with a transcriptional activator. So as a reminder, if you target this to a promoter region, you will initiate translation. Well, how can you measure that? So we used the doxycycline-inducible system for GFP. Normally, you have this transcriptional activator that only works in the presence of a tetracycline like doxycycline and will facilitate expression. What we did was we had DCAS9-VPR, a transcriptional activator, which targeted that same promoter region. Now, indeed, what we saw is doxycycline gives good GFP, but using either regular or these MS2-modified guide RNAs, we were also able to facilitate good GFP expression, which we could also measure as MFI. And then if we add extracellular vesicles, we saw this also work. So with a non-targeting guide RNA, we saw no increase. And with the targeting guide RNA, we did. And once again, CD9 strongly outperformed CD63. So being very happy with this, we thought, well, let's go for more clinically relevant systems such as base editors. And we also came up with a way to measure this. So our construct there is we have a red fluorescent protein stop codon on GFP. And if you make a single nucleotide mutation, change that stop codon into any other amino acid, you'll get a fusion protein of mCherry and GFP, and that you can measure. So we use an adenine-based editor. It turns A's into G's. So we did that on the opposite strand. So the A on the opposite of T, we turn into a G. And what happens then is that we get green fluorescence. So using the same dosage as we used before with Cas9, we did see some green cells, but the efficiency was very low. We spent quite some time trying to figure out what was happening here, and the issue was actually these RNA binding domains. Because these two stem loops, these two hairpins, stem loop 2 and the tetraloop, are the ones that we are binding with these MCPs. But stem loop 2 is very close to the enzymatic group that turns these A's into G's. So we thought, well, maybe there's steric hindrance, maybe it's in the way. So what we did is we expressed guide RNAs with either the MS2 loop only on the tetraloop, only on the second stem loop, or on both. First, we tested them in cells, and under normal conditions, everything works just fine. But when we tested these in extracellular vesicles, things looked very differently. So if we had any guide RNA that had this hairpin on the second stem loop, either by itself or on both, these are the gray bars, we saw very little gene repair. But if we only targeted the tetraloop, things worked incredibly well. So the conclusion is that this does work, but proteins still bound to Cas9, either through direct fusion, but in this case, by binding these RNAs, can interfere with the functionality. Well, of course, a reviewer asked us, well, show us something that's not just GFP, but an endogenous gene. So we picked a target on CCR5, where we could, by making a single point mutation, we could create a restriction site. So we treated these cells with our EVs, we isolated DNA, did PCR, and indeed we saw that we were also able to get gene repair. In this case, we're creating a specific restriction site with these EVs. So this is not just something that works on fluorescent protein constructs. But this does show that there is an inherent limitation to this approach, because these MS2 code proteins can interfere with functionality. So in a follow-up project, we thought, well, perhaps this is something that we can fix, because it doesn't matter what type of construct we put here that facilitates release, it's going to give the inherent same problem. What we need is we need to get these hairpins free from these MS2 code proteins. So we thought, well, perhaps we can do something about this interaction. Perhaps we can modify the affinity so we can still have loading, but there's opportunity for release. So the idea is as follows: if your affinity is high enough, you'll get a lot of loading, but if it gets too high, you're going to have issues with release. And most papers in the EV field are mainly focusing on loading, but your optimum is, of course, somewhere here. So we thought, well, perhaps we can see if this is true, and you can do this, of course, in multiple ways. One of the things that we looked at is there's a specific nucleotide in the MS2 hairpin, and if you make mutations there, you strongly affect affinity. So you have 4 options there, of course, C, U, A, and G. And there's also a few amino acids known in this construct. In this case, we focused on tyrosine 85, where you can also strongly affect the affinity. And so first focusing on the mutations in the hairpin, what we saw actually quite nicely fit our hypothesis. If we have this is listed in order of affinity. So if you have very high affinity, you don't get any Cas9 delivery. And if you go down in affinity, you do, until at a certain point you start to see diminishing returns. And the idea that we had is like, okay, here we've reached a point where we don't have proper loading anymore. But the question is, how do you study that? Because you cannot separately study either loading or release, just looking at these affinity-based mutations. So we thought, well, what if we bring back this photocleavable domain? Because in that case, we're only studying the effect of these mutations on loading, because with this photocleavable domain, you're going to have optimal release anyways. So doing that, things all of a sudden look very differently. And what we then see is indeed that these higher affinities perform incredibly well. So that means that there is Cas9 there. And you can actually go quite far down in the affinity without seeing any issues there. So if you have them side by side, I think it also explains nicely why this one specifically worked well, because this suggests that all these three have similar levels of loading. However, this one has the lowest affinity, so all of these three has the best release. And what's also interesting is that the wild-type hairpin is this one, the one with the U. And that works really well for the MS2 bacteriophage. It must have evolved that way, but for our approach, we actually need a lower affinity. So a lot of people just order their constructs from AdGene, but in this case, that's really not the best way to go about this. So of course we confirmed this. So Western blot analysis, we only saw actually at the lowest affinity that we had less loading of the protein, and we actually also saw that of the guide RNA, so that fits quite nicely. Now, like I said, you can also look at the interaction between the MS2 code proteins, and in focusing on amino acid 85, I should mention 2 of these MS2 code proteins interact with one hairpin, and this is not symmetrical, so basically it fits in 2 positions, so you need to make these mutations on both sides. Otherwise, you're going to have dual kinetics. And indeed, what we saw was the exact same pattern there. So this is the wild-type affinity. All our mutations actually lowered affinity, and if you put them in order of affinity, you'll get this similar pattern. So to finish off, whenever I show this work, I often get the question, well, okay, that's nice, but all good. Few minutes? I thought, okay, this is not the, I need to worry when you start throwing it at me, I'm guessing. Okay, well, I'll finish up quickly then. So people asked us, well, why don't you just make a direct fusion between your Cas9 either at the C or N terminus? Because that's so much easier, you have less constructs, you don't have to worry about all these affinities. And so we did try that actually as a comparison. So again, using this base editor construct, we compared a wild-type guide RNA to this MS2 guide RNA, and if you transfect them, they perform equally well. So what we did is we compared the construct that I just talked to you about to a direct fusion with CD63 either on the C or the N terminus. And the idea is, well, if the guide RNA runs into this, it will bind it as well. And that should also work. And it does, but it works around 100-fold less well. And this surprised us, especially because most people in the EV field are actually doing this. And so we thought, well, why does this not work so very well? So we started to analyze our EVs a bit better. And when we did single particle flow cytometry, we saw that we had quite some Cas9 protein in there, up to 10% of our particles. But only 1 in every 10 to 20,000 EVs had a guide RNA. So everyone's focusing on Cas9 loading. Guide RNA was the limiting factor. And I think that's why in so many papers, you see that people, to show how effective their system is, they add their particles to cells that are already expressing guide RNAs, because that is the main challenge. And I think that's why it's so important to focus on aptamers instead of using direct fusion to load your Cas9 into these types of particles. So to summarize, I hope that I've convinced you that Cas9 guide RNA RMPs can be expressed by cells and then loaded into EVs. You can do that in many ways. But in many cases, guide RNA loading and delivery is actually limiting. Additionally, yeah, you can use different fusion proteins. It really matters, specifically for the amount of loading you have. And I hope that I've convinced you that cargo release is at least as important as loading, and that fusogenic proteins can strongly increase functional delivery. This work has been published, so if you're interested in reading more about this, you can look it up online. I was specifically pleased with the fact that our affinity paper was published in the Journal of Controlled Release. I found that very fitting. And of course, lastly, yeah, this work was done by quite a few people. Specifically, we had 2 PhD students, Vivian and Omnia. They did a lot of work on this, but we're also incredibly grateful to all our collaborators. And with that, I'd be happy to answer any questions you may have. Thank you.